Okay, this is image analysis in earth science with more emphasis on images and less in earth science. So not surprisingly, I'm not going to teach you any geology because that would be just crazy. So what this talk is going to be covering is a little bit of why geologists need image analysis. And the main reason geologists need image analysis is that I can sell this book. So that's reason number one. Don't feel obliged to rush out and buy it. Image analysis, if it's a part of your project or research, then by all means get in touch with me. So where this came from, in a sense, but this particular talk I'm going to try and keep reasonably short because I'd much rather you have a little play with the demo table here rather than listening to me drone on for too long. So I'm going to talk a little bit about image analysis, about and how it allows us to study the microscopy of materials in terms of just the general principles. No details, no maths. You'll be glad to know, I'm sure. So, why? Well, no surprise that different geomaterials, regardless of what they actually are, so regardless of whether they are particles in a matrix or whether they are aggregates, the matrix, you simply see grain boundaries between exist in the rock, or whether we can't actually see individual grains, but we're still of the texture of the material. Regardless, there are processing so that we can not only get a better picture of what's going on, but hopefully quantify what's going on in these materials. For instance, we might want to understand something of how these things came to be. If we're talking about cataclastic rock, then we're talking about the processes, the physical processes such as fracturing and crushing and how to produce this structure. We quantify this structure and learn something about the processes that made this material. And similarly, for you might be interested in looking at the crystal structure, not only how large the grains are, but the actual circumference to learn about the stress and the strain that produce those structures. Of any materials that were formed by deposition, by studying the either the size of the particles or the relationship of the environment under which those rocks were put down in the first place. Processing and image analysis. Because this talk is mainly talking about those two things, we need to be clear about the difference. Image processing, where we start generally with another image, usually size, and the image is changed in some way to help. So image processing is a process whereby we generate a second, third, and that image in some way helps us interpret what it is. Generally speaking, it's either processed in order to help us. We don't see with our eyes, we see with our eyes and our brains, and so sometimes we need an image to be processed in order that we can look at it and extract more information. But quite often that image processing is the first analysis is when we take an image and we extract some image, some data. Usually it involves data and reducing it down to a much smaller set. So image processing generates another image analysis takes out some numbers depending on what it is you're trying to quantify. And it is very common that image processing is the first step in image analysis. So I'm going to be illustrating both of those with a few images. I'll throw a few other images in the time being before we get there. One thing you have to is not going back to the good old days of photographic emulsions. If we're talking about digital, as far as the computer is concerned, an image is just a collection, regardless of we're really just looking at a collection of numbers, and you can think of them in a sort of a two-dimensional grid. Like grade, we've taken a whole load of numbers and we represent them as shades of grey. Or if you take an image which has been digitized, each of the pixels has been converted into a number. But the computer only knows about numbers. So software will be built, either written by you or written by others, to extract some meaning from numbers, but the computer doesn't give a damn what the actual image looks like. Quite often, computer software will be instructed to look at differences of numbers, ratios of numbers. The actual number itself is often, it's usually that number means something. And if we've got a collection of, we can choose to represent that as a two-dimensional grayscale image where the gray in that two-dimensional collection of numbers. That's the conventional way of representing it. But we can find any color we like to use. We can have a grayscale value if we like, or we can just arbitrarily choose in any way we like. And we might choose to do that. The structure of that image, the structure of the thing, more readily available to us. And of course, we can usually see more by looking at images which are. Can I just check if blind here in the audience? Okay. Most people having a color image actually conveys more information because humans have grown up with a tricolour system of being able to detect red, green, and blue, and hence extract a great deal of information. But of course, we could shed load of ways. You're familiar with the idea of drawing contours. We don't image, but that's always an option. This particular image happens to be so the white blobs on the left-hand side are actually, and the grey of white in this case is an indication of how high these atoms are. Image that we're looking at. So these contours do actually make some sense in terms of so you're used to that idea when you're thinking about geological topography. Here we're talking about top. And if we wish, we can always represent that in this sort of way. We can map this to represent the topography of the surface, and we can ask the computer to represent that by imagining that light is shining from the upper right and casting shadows. Of course, this is still a two-dimensional image. It's simply a projection onto a screen that you're looking at. Your eye, and in particular your brain, will often interpret that as if it was a three-dimensional space. In other words, this is just a way of fooling your vision into giving a carefully shaded individual expression that is three-dimensional. Whenever we've got an actual speaking, the sample is we take it, whether it be with a camera or with a microscope, and then we end up with not necessarily the same resolution as the original. Of course, when we digitize it, it might be that we lose some resolution. And regardless of whether we display it as two-dimensional or give it some sort of pseudo-dimensional feeling, we have to remember that ultimately we are looking at a digitization of the original image. If we've got hopefully we don't lose too much of the original data that we were interested in. If we take an image, I'll explain where the colors come from shortly, but if we imagine that that top left image is our raw image, we can choose to process that from A and get an image that looks like B. That's, hopefully you see this as more of a three-dimensional surface. That probably looks quite flat, the image, but if we process it in a particular way, the main boundaries here, so that some of these appear to be higher and appear to be lower. Gray image and height, which gives you an idea of how to process this image. grains are aligned. And if we take that image and process it a different way, what's going on in the individual, what's going on in the grain themselves, and just highlight the grain boundary. It might be the grain that's interesting for us. So those are two examples of processing, but if we take that image and say, well, I know how big these grains are, how many grains of that size, how many grains of this size, I can end up with a distribution, a frequency distribution. And this is an example of image analysis where we've reduced the amount of data from the number of pixels in the image, and we've reduced it down to us at the time, in this case, perhaps simply a grain size distribution. So quickly, this, apart from trying to sell background, I started many years ago here in Liverpool in the Surface Research Centre over by physics, and I was interested in a particular type of scanning microscopy. I was looking at molecules, particular atomic arrangement of the surfaces of a particular set of metals, the rare table. And the technique I tended to use was scanning tunneling. With optical microscopy, you might have also dabbled with other types of microscopy. With scanning tunneling microscopy, a tip rasters backwards and forwards across the sample, and a current flows when a voltage is applied between the tip and the sample. And the current that flows, according to the rules of quantum mechanics, depends very sensitively on that gap between the tip and the sample. So even if that by half atom still easily be measured, and hence scanning tunneling microscopy is very sensitive to the atoms and the arrangement of atoms and molecules on surfaces. And there's an example taken a few years ago. If you can't quite read it, that scale bar there is 2 nanometers. So this particular image is a few nanometers by a few nanometers, and the individual mountains exaggerated for clarity. The individual atoms here are oxygen atoms in this particular surface, which is an iron oxide surface. There's actually a mixture of Fe2O3 and Fe3O4 in this particular surface, which is why the atoms are spread uniformly across the entire surface. There's little pockets of one phase buried in the other phase. Typical of what surface scientists are looking at the atomic level of surfaces to try and understand how atoms are arranged and how molecules interact with those surfaces. And this is a reminder of just how far you can go. This is not an image I've taken, but this image, you can see that the size of on a side is less than 1 nanometer, 850 picometers on each side. And if we were to imagine, well, if that's individual atoms, in this case, individual atoms of carbon, how big would a grain of sand be? Roughly speaking, the size of the moon. So, working with STM, I started allowed me and my students and postdocs, etc., to analyze images from various types of scanning microscopy. There are a large number of scanning microscopies in the family, some of which you might have heard of, some you might not. Scanning Auger microscopy, scanning electron microscopy, scanning pulse microscopes, scanning ion microscopes, scanning laser, scanning optical, and scanning tunneling I've already mentioned. And there are probably a dozen other scanning microscopes backwards and forwards and scan across a sample surface in order to generate images. And that's why this particular set of techniques is often referred to as SXM. You give me a letter of a particular scanning microscopy that it applies to. So all of these microscopies together are referred to as SXM by some people, including me, and I wrote software that allowed me to analyze scanning microscopy images. That software is now in use across a large part of the world, but I'm having trouble breaking into Antarctica, as you can see, the pins in those areas. And I spent a lot of time looking at and atomic positions. I wanted to understand where are the atoms in various metals, especially the rare earth metals. It's a three-dimensional crystal structure for each of the elements in the rare earths, and if you take three-dimensional various planes, you can then study to see where the atoms are. Are they where you'd expect them to be based on the bulk structure? And sometimes no. So that's my background, looking at scanning tunneling microscopy of various metals. But to show you how, imagine you had an image like this one in the top left. It doesn't really matter what it is. It happens to be a quasi-crystal. This is a crystal, but it doesn't have translational symmetry like crystals do. You can see it looks a bit of a mess. Some of the interesting atoms in the background, we can hardly see where they are. There's very large white blobs. They are contaminated. What would we do to process this image to try and make it a little more easy to see what's going on in an image like that? Well, because we think in this particular case this material has got, as indicated here, a tag symmetry, this plane, we could work out the frequency components. We easily, you've Fourier analysis and Fourier synthesis, but these days for you, you can simply ask the frequency components of that particular image in two dimensions, and you get a whole load of points. And if I point out some of them, you can see that they're actually. And the computer software that you're using, if it allows you to look at the frequency components, ask the question backwards. Show me which parts of the grid have got symmetry, and throw nothing symmetry. So you can effectively do the backwards, and then you can see you get a much cleaner image. It's effectively the same image, but it's now got none of that everywhere because they to do with tenfold symmetry, and we've now got a much clearer indication of how the atoms are arranged with this. We have to, whenever we're doing any processing, we have to be careful we don't end up generating an image which bears no resemblance whatsoever to the original image that we're trying to process. And in this case, we can do a quick double check. For instance, you see there, atomic arrangement appears to be a sort of sunflower. There appears to be a ring and 10 atoms, or at least 10 objects, which are atoms, circling around. Is that actually how these atoms are arranged? Well, if you look at exactly the original image, you can perhaps just make out that it's rather low contrast and rather scrappy, but yes, there is a ring with 10 atoms arranged in a circle. Which makes that so much because we have effectively removed everything that wasn't of interest and that is quite often what we do with image processing, try and separate the wheat from the stuff, throw the stuff that doesn't interest us. And I spent some time in the 90s and I guess the early noughties looking at images and looking at, in this case, trying to understand how those molecules were arranged, seeing if I could recognize whether there was a particular pattern to how a particular molecule wanted on the surface. It was of interest to, for instance, catalysis, understanding how molecules sit on semiconductor surfaces affect their chemical properties. In this case, it didn't look to be a particular pattern. Similarly, looking at a set of molecules sitting on an atomic surface, which appear at first sight to be in a simple grid pattern, but when you look more closely at the grid, they're sitting slightly off, and some atoms sit quite close to the grid. Why should that be? Well, in this particular case, that was part of the investigation of a PhD student to actually try and investigate, see what was going on. And on the right-hand side, it might be a test image, or if you like, a false image, a regenerated image, in which I've drawn a circle for each dot here, and I've color-coded it according to position very close to the grid or a long way from the grid. Any atom which is in close to the grid points is blue. Any atom which is sitting a long way from the grid points is red. Again, using color color here because I know that humans with normal color vision can interpret it like this and see red and blue much more readily than you can look at the original image and see, well, that little splodge to the etc. So processing it in this way, and in this case color coding the result and interpret it more readily. But we have to be careful, because whenever we're indeed processing an image, we have to be careful about visual perception, because what we see when we look at an image can often be very different from what is actually there. And I will show you that in this case, because we're going to ask the question, which is better, your brain or a computer when it comes to image processing? In other words, a computer made of carbon or a computer made of... And the answer is, it depends on the problem. In some cases, computer, in some cases, human vision can be fatally flawed. I found a post on images on whether or not that by color, but I'm about to show you you cannot rely on your color vision. You can't rely on your color vision. So you all see a cube on the left and a cube. I'm interested in the tiles 5 by 5. There are 25 tiles there on the left. How many of them are blue? Audience participation. I saw a 4 in the background. Any other numbers? 4. I'm not sure you 8, but okay, 4. These. I would think advocate here, Richard. Okay. That blue means these things. So it's 4. I think everybody said 4. 10 picture, same question. We're only interested in the 25 tiles on the top there. Right, 4 blue, 7 yellow on the right. How many people are prepared to put money on that? There were 4. 6 yellow. Most people would say 1, 2, 3, 4, 5, 6, 7. 7 yellow tiles on the right. But that is actually not correct. Because what I can show you is, and I'll do this twice to convince I'm not lying, it looks like trickery, but this is not trickery. I am going to remove all the pixels other than what you think are the 4 blue tiles on the left and the 7 yellow tiles on the right. I'm going to take all the other pixels away, and I'm going to do that one step at a time. Those are the tiles you were looking at. The tiles on the left, they're grey. They're actually the same color. Again, now convinced, no, they're not grey, they're blue and yellow, or bluish and yellowish, depending on your level. But this is a reminder that you cannot judge color. Color differences. You notice that the background, of course, here is yellow, and the background here is blue. That is putting on doing image processing. It has to, because there are millions of receptors in your eye many times per second. You cannot process the information. Your brain is always making shortcuts for you because it thinks that's what you want. And in this particular case, it's saying those 4 tiles, well, they're actually bluer. This side, that your brain is telling you that these appear to be yellow, actually they are yellower than all the others. And if we move those pixels, then we find they're actually grey. Here? Yeah. Yes. I haven't changed any of the pixel colors inside those 7 and 4 tiles. And it's difficult to believe. There's two ways you can convince yourself. You can go to a web page and convince yourself that it takes punch up, then hold the paper such that you can see, for instance, one of these tiles, but you can't see all the others. Effectively, hole so that you can only see one of these. You can almost do it by, if you can get it down to the point where you can just see that one tile, a bit tricky depending on your fingers, you might be able to convince yourself that is not blue, it's grey. And if you do the same over here, you'll convince yourself that's not yellow, it's grey. So I suggest you go to lottolab.org, assuming it's running. It's a reminder that colour is not absolute, it's all relative. So if you have a rock and some nice orange, and this one has almost, you cannot judge the colour without being very careful. You can be fooled. So if you want to know what colour it actually is, get a computer to tell you rather than judge it by eye. And you might say to yourself, okay, well, yeah, but that's a little bit, yeah, that's colour. But I can tell the difference between two shades of grey. I can tell them rock. Well, have a look here. Have a look at the two squares A and B, not the A and the B letters, those are sitting. So we have a square up here labelled A and a square there labelled B. Which one? You're now very... Yeah, stop it, Richard. You're cheating. Most people would say, well, obviously, square A is darker than B. It's pretty obvious. Let me do the same again. I'm not going to touch A and B at all. All I'm going to do is bring in a blank set of pixels from the left and a blank set of pixels from the right without touching the shade and on the right. Exactly the same. On and B, the square labelled A and the square labelled B are exactly the same shade of. Now you absolutely know grey. What's your brain? Can you force your brain having grey to see them as the same shade of grey? Generally, you can't. You can't override what is a million years worth of image processing that the brain has learnt to carry out. It is intrinsic to our evolution. To help save you getting eaten from something behind a bush. Processing is something that happens all the time, and it doesn't matter as long as you're aware of it. If you're looking and using your eyes, about what it's looking at, whether it be colour, whether it be those flecks, bright, those, be careful. You can be fooled. This is the opposite. You think, well, okay, I think that a computer should be able to deal with this quite easily. You look at this and you say to yourself, what's going on? Okay, this is an image of DNA molecules on a glass substrate. The glass, although it's polished, is actually rough on a scale. So you get this rather mottled grey background. But these white objects here, these stringy little objects, these are the DNA. If I asked you how many DNA strands have we got there, well, I don't expect you to do that, but if I gave you a couple of minutes, you could probably work out how many were. Let's say, for the sake of argument, 25. You ask yourself, well, if I can work out the 25 DNA, it's easy to do the same thing, given this image. Actually, no, it's a rather tricky problem for a computer to work out. You look at this and you say, well, all the DNA strands are brighter than everything else. If bright means high, the surface. That should be easy. But if we actually take a line, I'm going to tell you what it is. If we take the intensity variation, we would expect it to look like that. Surely a little bit of, and every once in a while we're going to slice through a DNA, and that's going to be a lot background. That's what you're sort of telling you that this image is, but actually that's not what a profile through and plot the intensity as a function of position. It looks more like that. It is an absolute fraction, and it happens to be that one there. So you can ask yourself, well, if that's glass, 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 and then I am going through a DNA molecule and then another one, which of these peaks corresponds to going through the DNA molecule, and which of them is the background glass? Brain is telling you the background glass is a grey and the DNA is white, but actually that's not the case. Height here in nanometers corresponds to grayscale. So in this particular case, we're going from the left-hand side of this profile is at the top, and the 3 strands of DNA are cut here. So everything on the left-hand side is glass, and somewhere in there, there are 3 strands of DNA which we're cutting through. And they are actually that one, and that one. How on earth your eye and your brain fix it out immediately. No problem. You ask the computer to pick out which of these is DNA. That is a rather tricky problem. To actually solve that, I found, was not to work on the original image, but to such a way that I made it obvious where the DNA is. In other words, I processed the image until I got something that looked like that. Now, the DNA is obviously as black, and everything shows itself as white. So now I can get the computer to work out how long these are, how wiggly they are. Easy once we've changed the image into something that the computer is a lot happier with. The computer is much happier with that than it is the original. You look at it, you think, what's the problem? What's going on in this particular case? So it's processing. Sometimes easy, sometimes quite tricky. Whenever you're looking at images, be careful, they can be quite tricky. Let me say a little bit about the nature of light so we can use microscopes to look at materials, ultimately Earth materials. So James Clerk Maxwell says that light is an electromagnetic field. So light propagates with an electric field and a magnetic field. And we can imagine they're both sinusoidally varying. We can imagine, for instance, the electric field, which way have I got it in this particular case? The electric field is going backwards and forwards, left to right, and the magnetic field is going up and down. And we can see that in this particular picture where I've explicitly said, in this particular example, the electric field is horizontal and the magnetic field is vertical. But whenever we're talking, we know the magnetic field is there. Need to say field pointing, field oscillating in a something. We don't know what the magnetic field is doing as well, because we know the magnetic field is always at right angles to the electric field in an electromagnetic way. Simply don't draw it. So in this particular model, long, I'm not claiming that. Because there is no need, because we know it's there, so we don't worry about it. And when we talk about the polarization vertical, if we're talking about horizontal polarization, we mean the electric field is oscillating in a horizontal. The magnetic field will always be there, but let's just forget about it because it's not worth talking about. One thing that I'm not going to say much on the front table here is there are other ways of manipulating the polarization of light in a way that I'm not going to talk about. It's possible to combine linear polarization of light and actually end up with light that is corkscrewing. Rather than terms of the electric field pointing up, pointing down, pointing up, pointing down. By combining it with another linearly polarized light beam, it is possible to go up and down, but to actually call clockwise or anti-clockwise. And although you never expected to find circular, you down have a close up of those shortly. So let's actually look at get information about Earth using if we take a sheet of through light polarized in a particular plane, for instance, if we let through light that's only polarized represented by the red arrows, we then take another polarizer, position it the other way round, so it only lets through light polarized vertically. So no light can, because light is polarized one way, and it's only horizontal for the moment. If we place them like that, no light can get through. But what happens if we place a sample between the two? Now the sample in the Earth material has perhaps a section of Earth material, section of types of material. Here I'm using a different type of example. I'm using a section which is not all the materials. I'm using the same material of different thicknesses. And that illustrates the same idea, but without actually using an Earth material section. So we're going to look at cross polarizers, which shouldn't let any light through. And we're going to see what happens when we place a bit of plastic in between them. So let's switch over to the camera. So the camera is looking to my right, and might help perhaps if we have lights down. Let's see what happens. Okay, as well as picking up a little bit of clutter on the bench, the important thing is that we're picking up this black rectangle, which is two polarizers. Now, a little bit of light is leaking through because these polarizers aren't perfect, but if I could get perfect polarizers, very little light would be coming through. Perhaps if I just turn them away, you can see the light is actually quite bright, so just a little bit of light is leaking through. And this particular sample, if I just put it on again, this particular sample, I think you'll agree, is transparent. If you look at my shirt, you can see that there is no color in these bits of sellotape, or these bits of plastic. There's no color there, and it's almost transparent. But what happens if I take this piece and don't place it in front or behind the polarizers? What if I place it in the polarizers themselves? Let's see if I can make the best possible use of this. Let's try it that way around. So I'm now going to carefully place the plastic in between. And the camera has a bit of trouble adjusting. But then, after a little while, I hope you see colors. And the colors will depend on the thickness of the sellotape on this bit of plastic. Let me just let the camera adjust again. Perhaps I can get it to stay there. So can you see that we've got lots of bits of sellotape and different thicknesses of sellotape, where sellotape sections are overlapping each other, give us different colors. So how is it that if I didn't have those between the polarizers, I would effectively get nothing, but as soon as I insert it, light comes through? So what's actually going on here? Let's switch back to the PowerPoint. That's effectively what we've got, a picture taken under slightly better conditions. Let's have a little bit of background light. So we see very little of the sellotape in the background, except when we're looking at it sandwiched in between two crossed polarizers. What we've actually got here is this. We've got a light source which is effectively unpolarized, generating packets of light, photons that are coming out at random, sometimes vertically, sometimes horizontally, some intermediate angles, polarized light going through a polarizing filter, which ends up with the electric vector vertical. Then it's passing through what could be called an optically active material. Now, in my case, that's a piece of has been extruded and the molecules have a particular direction to them. When you're looking at a mineral section, you're looking at which they interact with the polarization of the light. In other words, coming through an optically active material is not the light coming through in a number of different ways depending on the material we're talking about. Looking at different in directions, but the upshot is material when an electric vector that is vertical passes through a material, whether it be a sample, whether it be a piece of sticky tape, it can change the polarization. So in this particular example, the polarization appears to have been rotated through a 5 angle. How much does it rotate? That depends on the distance in the material. In my case, all the same amount of material, same material. Looking at the mineral section, probably all the same thickness, but different minerals will rotate the polarization by different amounts. And remember, none of the light will get through if the light is vertically polarized, because this, it says analyzer, but really another polarizing filter. That light won't get through if the vertical nature. We now have a horizontal component, polarization. With a horizontal component, some light will get through. It's often of the wavelength of light used. In this case, we're using white light. All colors are present, and the amount by which the light gets rotated will depend on the wavelength, and hence you get different colors showing up for different thicknesses, or you will see different colors corresponding to the sample. One extreme example of that is calcite. The light traveling through the calcite, the light will actually travel at a different speed one way compared. So it's not simply that the polarization at which the crystal differ, and you could get a situation where light can take two different to the crystal. Is polarized one way, the other image is polarized the other way. You to polarize the filter to show that that is the case. Although you see a double image, one from the other comes from that. You can prove that for yourself at the front. So let me finish. I don't want to go on too long here. So let me say a little bit about microscopy of materials. I'm sure you're itching to come and look at these beetles on the front bench here, so I'll just briefly say, looking at samples, we might be through cross-polarizing microscopes. It's also possible that you're looking at a polished surface of a particular material. I'm not sure if these days you still use acetate foils in order to get a dimension looks like, because remember, if you polish a surface and then look at it in detail, depending on the matrix of the material, looking into the material, and you might be seeing some things that are actually below the surface. Whereas if you take what's going on the surface, so you get a true two-dimensional image of what's going on in that particular material. The analogy I drew earlier, piece of sellotape, which is essentially transparent, dirt and thumbprints, essentially transparent. Well, geomaterials are essentially transparent as well. You make them in very thin sections. But if you put them through filter, either a surface polarizing filter or crossed polarizers, and then take an image with a mono camera, you're looking at the geomaterials. One popular is to send the light through cross polarization, shown here, and then pick one particular wavelength of light that is transmitted, and that gives you information that allows you to quantify not only what you're looking at, quantify crystal structure, relative to the polarized beams. Remember that this down here, these are the colors that you might see if you were looking through a polarizing microscope. Don't confuse that with taking a grayscale image, saying I want to reassign each of these grayscales to some other color. Sometimes it's more easy to see the difference between the shades of gray of two adjacent grains by literally just making them different colors. It's easy to look at such an image and extract information from it by taking a grayscale image and then just arbitrarily coloring the grains. Useful is a situation where the coloring isn't somewhat arbitrary, but is actually giving you information of atoms within the grains that you're looking at. It might be, when you look at it might be that each mineral in your section is coming out as a nice different shade of grey, such that if you look on there, of shades of grey this way, or intensities of pixels versus how many pixels that. In some cases, you find that they are very nicely delineated. Okay, there are some overlaps between these distributions, but effectively by picking pixels within a particular range of grays, you can pick out that is this, that, or the other. I won't go into the actual materials here. You might notice that there is some overlap. Some pixels occur in more than one of these subgroupings, because these distributions are not totally different. There are some pixels which occur in each of these ranges. 100% segmentation simply from the level of grey that you've obtained from your images. But when you can see individual grains and you can see individual grain boundaries, then of course image analysis allows you to pick out structures to say, well, it's a, but it seems to be really long, it's wide, and it's oriented at this angle. And when you look at a large number of these grains, you can start to say, well, anything sort of necessarily circular, but is there any indication of elongation, a preferred direction in which they sit, which will tell you about the history of this particular sample. And of course, you don't need to do anything manually. Once you have reduced your image to a collection of structures, then that's the sort of thing that computers have for breakfast. You wouldn't want to go through this manually, saying this one appears to be oriented that way, this way. You can simply throw this at a software, an image analysis package, and it will simply calculate the area and the position and the length and the width and the angle of which this seems to fit. It will do that for every image that it sees, every object that it sees within the image. And given that, you can work out the mean azimuth orientation, and of course you can then generate any number of you want, whether it be the size distributions. Height doesn't necessarily apply. Doesn't apply if you're dealing with thin sections where each of the minerals you're looking at are by definition all the same height. But if we think about what we can get if we have images like this, we can put them through polarizers or circular polarizers and pick out the between individual grains, which we can reduce from an image to something which is basically just a map of where the grains are and how they're distributed. By eye, we look at the one on the left and the one on the right, what is white and grey. Look at this and we say, well, by eye, I am pretty sure that the grains on the right are smaller than the grains on the left. But one thing that's difficult without some computer software to help them is that if we ask, well, what's the most common grain size on the left and what's the most common grain size on the right, turns out it's actually the same for these two. If we ignore the bottom, the white and the grey, the most common grain size is what, between 30 and 40 microns in this case, and it's also between 30 and 40 microns in this case. The most common is the same as those two. That would be very difficult to pick out by eye because your eye is misled by the fact that there were more smaller grains in one case, more larger grains in one case, whereas this case the grains tend to be smaller. So the most common grain size, average, or the medium grain size, can be quite different, and your eye and your brain are not to do that sort of analysis. You have to rely on software to give you that information. And similarly, here we're looking at, I think it's a quartzite sample, which is being stressed, and so we're looking as a function of time, A to B to C to D, as this particular stabilizing. You look at that and you say, well, it seems obvious by eye that as we go from A to B to C to D, it's obvious that the crystals are getting smaller. But again, you have to be careful, because that when you ask what is the most common grain size, 3 and a bit, and 3 and a bit, and 3 and a bit, the most common grain size doesn't actually change, even though this. So, depending on what information you want to get out of these images, are you interested in the mode? When you look at these images, your eye is good at that, and you have to find where to give you the numbers. Having said what we saw a moment ago of looking at samples come from the material with light, here's one example where we've reduced it to grayscale, reduced to the boundaries are so that we can get the grain sizes, and then purely we've chosen to say, now that we know what the grains are, let's color them according to their size. In other words, let's make the large grains red, let's make the small grains blue, and let's make the medium-sized grains green, because most people looking at those two find it easier to say, well, there's a lot of red in this one, so clearly there's a lot of larger grains, and there's a lot of blue in this one, so clearly there's quite a lot of small grains in it. Allowing the eye and the brain of humans to interpret what you're looking at. So try interpretation. These are not the colors you would see if you put these samples into polarized. Final point about what can be done, rather than just banging something in between colors, what you can do is, with a mono camera picking up in particular wavelength, you can put a sample between cross polarizers and then take a picture every time you move the sample by 10 degrees. Of 18 images at 10 degrees from 0 to 180 degrees. Why should that be of interest? Well, if you look at grain number 3 there, it starts out quite dark, and as we go through this deck of cards of different angles, we see it gets lighter. Whereas grain number 2 starts off a middle grey, gets a bit lighter, then gets a bit darker. What happens to grain number 1? Well, it starts off mid-grey and it looks like it stays mid-grey. Why should they all behave differently? Well, this is a result. I can't illustrate it with sellotape because it doesn't work with sellotape, but here the alignment of the actual atom arrangement are not necessarily all pointing the same way. And so if the sample is horizontal, in some cases the arrangement of the axis, it looks pretty much the same regardless of the horizontal. Other grains in a particular sample might have their, in which case it does make, and so you and the incoming polarized light. So by monitoring how these intensities change as a function of swinging through the angles there, we can actually extract information about the three-dimensional arrangement of the crystal relative to the microscope. And that is a principle called computer integrated polarization. It's taking the idea of putting objects between cross polarizers, in some cases using circular polarizers as well, and actually extracting information about not only the polar angle, but the angle of a particular crystal relative to the microscope slide. So by taking a whole number of images, it's then possible to map out what is the for each pixel, ultimately each grain in the image. And you can tell the difference between one grain and another simply because they are misaligned to the difference. But because we're mapping it out over many, many images, we see alignment between misaligned a few degrees. We can map out grain boundaries, even those that are actually quite difficult to see. We can map out differences in orientation. And again, in this final grain map, not real colors. In this case, we're coloring them according to which direction a particular crystal is arranged. In other words, if the crystal happens to be arranged, for example, the symmetry axis is vertical, it would be approximately white. Whereas arranged in a particular direction, which we call north, then it would be colored red. If it was arranged in the opposite direction, it would be colored cyan. If it was tilted to the left, green. If it was tilted to the right, color it whatever, bluish. So we pointing clear sky, if you like, and there we can see that some of these are very similar colors, but they are very slightly different, which is why we believe there's a grain boundary between them. So that is how we can use the idea of samples in between polarizing filters, just putting it in a that's where the grains are. But if we wish, we can go beyond that and start to actually delve into the crystal orientation of grains and extract yet more information. So I've talked for long enough. I've given you an idea of what image processing and what image analysis is all about and how it. But I invite you to come down here and ask questions as well if you wish, and actually see for yourself some of the aspects I've been talking about. How vampires don't occur sight and double refraction, and I'll explain what I mean by these bugs here. Thank you.